What's up, Bio Penguins? Today we're going to do 2019 number 3. This one has to do with cellular respiration, specifically looking at the PDC. Okay, so it goes in, it talks about pyruvate dehydrogenase complex, PDC, catalyzes the conversion of pyruvate to acetyl-CoA, a substrate in the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals' PDC deficiency, a rare disorder. They first want us to identify the location of where PDC is most active. So it tells us in the prompt that PDC is going to catalyze the conversion from pyruvate to acetyl-CoA. So this is what a lot of people either call pyruvate oxidation, or they call this the link reaction or an intermediate step. Whatever you want to call it, doesn't matter. But what is important to note is where this takes place. Well, this is going to take place in the mitochondria, specifically in the mitochondrial matrix. So you could have put either of those options there, mitochondria or mitochondrial matrix. So let's see what the student put. PDC is most active in a cell's mitochondria. So of course, they receive credit for saying that it was taking place in the mitochondria. Part B says to make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by Krebs cycle in a cell. Provide reasons to support your claims based on the position of the PDC catalyzed reaction in the sequence of the cellular respiration pathway. So first thing, let's think. What is NADH for? Well, NADH is used as an electron carrier to bring the electrons from glycolysis in the Krebs cycle to the electron transport chain for us to be able to pump protons against the gradient into the intermembrane space, and that gradient then is used to make ATP. So what we're asking is, how does the deficiency of PDC affect those carriers? Well, number one, glycolysis takes place before the link reaction. It takes place before that step that the PDC catalyzes. And so since it takes place before that step, we're going to find that there's no change in the amount of NADH that is produced by glycolysis. Okay, so glycolysis will still continue. The PDC is not needed, or this takes place, glycolysis occurs before the conversion of the pyruvate to acetyl-CoA. So what did the student put? Oh, I'm ahead of myself, sorry. So in terms of the Krebs cycle takes place after this link reaction or after the pyruvate oxidation, and it requires the acetyl-CoA. The Krebs cycle, of course, starts with acetyl-CoA. It completely breaks down all the carbon dioxides, releasing out the 2 carbon dioxide, since acetyl-CoA has 2 carbons. It also makes 6 NADHs, 1 ATP, and 1 FADH2. And so they want to know, okay, well, what happens to the amount of NADH? Well, since this takes place after the link reaction, we're going to see there is a decrease in the NADH. So, of course, we see decrease. Krebs cycle is greatly reduced or slowed because there's less acetyl-CoA. And then this step is going to take place after the pyruvate to acetyl-CoA conversion. So the student said that there was a PDC deficiency, does not change the amount of NADH produced by glycolysis. They go on to then say that this takes place after glycolysis, and that's why we don't see that change. The second point they would have gotten would have to do with that it decreases the amount of NADH in the Krebs cycle, because without the acetyl-CoA, the Krebs cycle cannot occur, and that the PDC deficiency would halt all of that production at that step. And the last part of this question, the PDC deficiency is caused by a mutation in a gene, is located on the X chromosome. A male with a deficiency and a homozygous female with no family history have a male offspring. We have to calculate the probability that the male offspring will have a PDC deficiency. Well, the first thing we need to do is we need to think about, well, what does it mean to be on an X chromosome? Well, that's called sex linked. And so it's going to be found on the X chromosome. And so the male is your XY individual and your female is your XX individual. And so they're saying that the male has the mutation on its X chromosome, while the female has no mutation on their chromosome, which is why it's called homozygous, because of the fact that there is the 2 of the same allele. And it says no history, which means that there is no allele for the PDC in the female. And so if you were to do a cross between a female and a male, you're going to find that the only way to get a male offspring is for the male to pass on the Y chromosome, which means that his faulty X chromosome, his X that has a mutation, was not passed on. So there's a 0% chance that the male offspring is going to end up with this trait. So the options are the probability of inheritance is 0. The offspring will not have the PDC deficiency. So the student went on, they did a beautiful little Punnett square here showing that where the mutated X, and here you can see that the females were the only ones that are carriers now for it, and the male does not have that trait. So they said that there is a 0% chance that the male offspring will have a PDC deficiency. So I hope that was helpful. And remember, if you've had a pain, just success. Bye, y'all.